The overall goal of the following experiment is to characterize the local mechanical environment of lung parenchyma at a spatial scale relevant to resident cells by directly measuring the local elastic properties of fresh murine lung tissue using atomic force microscopy microindentation. This is achieved by cutting the agarose inflated mouse lung tissue with a razor or scalpel blade to prepare lung parenchyma strips of 5 by 5 millimeters in length and width and 400 micrometers in thickness. Next, unfixed, unpermeabilized lung tissue strips are immunostained, which identifies areas of interest for AFM microindentation. Then, AFM microindentation is performed on tissue strips in PBS at room temperature in order to directly measure the local elastic properties of the fresh lung tissue. Results are obtained that show striking differences in the range and distribution of tissue stiffness in normal and fibrotic lung parenchyma, and large spatial variations in stiffness, particularly within the fibrotic lung sample, based on stiffness maps extracted from force displacement curves obtained using AFM microindentation.
The main advantage of this technique over existing methods, like tissue strip stretching, is that it offers unprecedented spatial resolution, providing a unique perspective on microscale variation in tissue stiffness. This method can help answer key questions, such as how stiffness varies spatially within the tissue, and what the scope and the spatial scale of the stiffness changes are in the disease processes that result in tissue remodeling.
To prepare strips of lung tissue, begin by stabilizing the lung structure for cutting by inflating isolated mouse lungs intratracheally with 50 milliliters per kilogram body weight of warm 2% low gel point agarose prepared in PBS. Tie off the trachea and cool the inflated lungs in a bath of PBS at 4 degrees Celsius for 60 minutes. The agarose will gel and stiffen in the air spaces to gently stabilize the lung structure during this interval. Using a razor or scalpel, cut the agarose-stabilized mouse lung tissue into strips of approximately 5 by 5 millimeters in length and width and 400 micrometers in thickness. Then wash the strips in PBS at 37 degrees Celsius for 5 minutes to remove residual agarose. To exclude large airways and vessels, cut strips from subpleural regions distant from main stem bronchi. If airways and large vessels are to be imaged, cut strips from lung tissue more proximal to main stem bronchi. To isolate areas of interest for AFM microindentation, visualize the tissue by phase contrast microscopy, or immunostain and visualize by fluorescence microscopy. Please see the written portion of this protocol for the procedure. Immediately before AFM characterization, attach the tissue strips to poly-L-lysine-coated 15 mm cover slips by raising the cover slip from below the floating tissue, making sure the tissue strip spreads evenly on the cover slip surface. If necessary, sandwich the strip with a second clean, uncoated cover slip and apply mild pressure to assist with tissue attachment to the poly-L-lysine-coated cover slip. Calibrate the AFM system following the manufacturer's instructions immediately before each round of microindentation experiments. For this, determine two critical parameters: the cantilever spring constant using the thermal fluctuation method in air, and the cantilever deflection sensitivity, which is a parameter used to scale the photodiode output signal to the actual cantilever deflection distance. Calibrate the deflection sensitivity by obtaining a standard force displacement curve in PBS on a clean glass slide. Then calculate the slope of the force displacement curve. In the force displacement curve measurement, the AFM tip is extended towards and retracted from the sample surface at a single location, with the deflection of the cantilever, delta D, monitored as a function of tip displacement, delta Z. It is recommended to use a silicon nitride triangle cantilever with a 5 micrometer diameter borosilicate spherical tip. Using AFM probes with a spring constant of 0.06 newtons per meter, we have mechanically characterized soft materials with shear moduli spanning 100 to 50,000 pascals. Wipe the bottom surface of the sample cover slip with tissue paper, then fasten it to a standard glass slide with vacuum grease. Mount the glass slide on the AFM sample stage and cover the tissue with 500 microliters of room temperature PBS. Then place the AFM scanning head over the sample. Adjust the microscope sample stage to set the microscope for eyepiece viewing to align the AFM tip in the center of the field of view. Move the AFM sample stage to choose an area of interest on the tissue. Then switch to CCD camera viewing to record phase contrast image and/or fluorescence images of the tissue as desired. Move the AFM tip slowly downwards until it is in contact with the sample. Accurate AFM microindentation characterization of soft samples requires small indentation depths to avoid large local strains, which invalidate the Hertz model used for elastic modulus calculation. To avoid large strains, perform indentation in trigger mode by setting the cantilever maximum deflection to 500 nanometers. This deflection limit will restrain the maximum indentation force to less than 30 nanonewtons. The indentation velocity should be selected to be sufficiently slow to explore elastic rather than viscoelastic properties of the soft sample. Select a velocity range of 2 to 20 micrometers per second for lung tissue. For a single measurement from an area of interest, move the AFM probe to the location of interest and perform a single indentation to collect a standard force displacement curve. The probe will move only in the z direction. For automated mapping of a region of interest, switch to force map mode. Select the scan size and sampling points within the selected area. The AFM tip rasters across the sample surface between indentation movements and collects individual force displacement curves at each point within a defined sample grid. We found it practical to use a 16 by 16 sample grid to map an 80 micrometer by 80 micrometer area at an indentation velocity of 20 micrometers per second, which can be completed in about 10 minutes. To calculate the Young's modulus, fit the force displacement curve to the Hertz spherical indentation model using least squares nonlinear curve fitting as shown here, where F equals K sub C times delta D is the force to bend the cantilever, K sub C is the cantilever spring constant, R is the sphere tip radius, delta equals delta Z minus delta D is indentation, and nu is the sample's Poisson ratio, which equals 0.4 for lung tissue. To assess the quality of the fit, calculate the SSR value, or the sum of squares of the difference between the data and the fit values during the nonlinear curve fitting. Eliminate unreliable or uninterpretable measurements by discarding data from bad curves with large SSR values. If desired, convert elastic modulus, or E, to shear modulus, G, using the relationship E equals 2 times the sum of 1 plus nu times G. To visualize spatial patterns of stiffness collected in force map mode, plot modulus data in contour maps, for example in a 16 by 16 grid covering an 80 micrometer by 80 micrometer area. To process a large amount of force curve data, a custom algorithm may be written to automatically fit force displacement curves, extract moduli, and/or plot contour maps or elastographs using the same procedures and parameters just described. This figure shows that in properly cut and stained tissue, the alveolar microarchitecture of lung parenchyma is well preserved, as observed by immunofluorescence staining for the basement membrane component laminin, without fixation or permeabilization. These panels show immunofluorescence staining for collagen 1 in fresh, unfixed lungs harvested from mice previously treated with PBS or treated with bleomycin to induce fibrosis. Sample force displacement plots obtained from AFM microindentation are shown here. With the same applied force, the AFM tip generates a large indentation on a soft region, resulting in a relatively flat force displacement curve, shown in blue, versus a small indentation and a steep force displacement curve for a stiffer region, shown in red. In order to obtain clean force curves, after each indentation, the AFM tip needs to be retracted completely from the sample surface and free of contact before the next indentation. This contact-free state corresponds to the flat region of the curve, where the tip translates without cantilever deflection. This figure shows a typical false curve without a flat region, which occurs when the tip is not completely retracted from the sample surface, such as when soft sample attaches to the tip, making it impossible to determine the contact point. If the tip is completely trapped in soft sample, the curve may look like this, without clear deflection, but small noise. This figure shows stiffness data extracted from force displacement curves displayed spatially as stiffness maps, referred to as elastographs, where color scales with stiffness. Elastographs demonstrate striking differences in the range and distribution of tissue stiffness in normal and fibrotic lung parenchyma, and large spatial variations in stiffness, particularly within the fibrotic lung sample.
After watching this video, you should have a good understanding of how to characterize the local mechanical environment of lung parenchyma by directly measuring the local elastic properties of fresh lung tissue using atomic force microscopy.